Insulin is synthesized in significant quantities only in beta cells in the pancreas. Elevated concentration of glucose within the beta cell ultimately leads to membrane depolarization and an influx of extracellular calcium. The resulting increase in intracellular calcium is thought to be one of the primary triggers for exocytosis of insulin-containing secretory granules. The fusion of insulin granules with the cell membrane can be modulated by calmodulin phospholipid or CMP-dependent protein kinases. Release of insulin can be also influenced by several classes of protein phosphatases, including the calcium or calmodulin-dependent enzyme, which can be called calcineurin. The principal functions of kinase and phosphatase enzymes is to target protein kinase and phosphatase to cellular microenvironments where they have access to particular substrates and to segregate their binding partners in a manner that prevents the indiscriminate relay of signals. I-kinase anchoring proteins are highly organized molecular complexes determine the precise location and timing of the signal transduction that occur of a cell surface receptor activation. It's also a group of multivalent binding proteins classified on the basis of their ability to interact with CMP-dependent protein kinase. One anchoring protein of particular interest is AKAP79 or 150 that targets enzymes including PKA, a guanine nucleotide exchange factor, and protein kinase C to the membrane. This particular anchoring protein could be a potential modulator of glucose homeostasis, as previous studies have shown that this anchoring protein is functionally coupled to G protein coupled receptors, adenyl cyclases, and ion channel.
According to the materials and methods of this research, the first and most important to mention is the animals used. In this experiment, mice specifically were housed at the University of Washington under a 12-hour light and dark cycle with free access to food and water. Now, according to ethics and ethical issues, the institutional IACUC review process approved all these procedures before the experiment. Furthermore, the researchers used various methods like cell culture or immunoprecipitation and Western blotting and tissue analysis involving many buffers such as RIPA for immunodetection or phosphatase inhibitor buffer. This process was performed for 2 hours in 4 Celsius degrees in the presence of 25 microliters of agarose beads, 2 microgram antibodies, and 1 milligram of lysate. The output information of this research is very interesting and may be extremely useful in future drugs. Alpha kinase anchoring protein 150 is an important protein because it basically organizes insulin secretion. Membrane depolarization and the concurrent mobilization of calcium plus 2, but also the CAM signaling cascades drive the regulated exocytosis of insulin granules. Basically, this protein makes an impact by integrating with signaling pathways and probably creating a voltage in a whole cell patch mode, which therefore increases the peak current densities and the secretion of insulin.
Surprisingly, AKAP150 knockout mice showed improved glucose utilization. The authors tested protein kinase B, which is an intracellular mediator of insulin and glucose transport, and found that protein kinase B signals to be amplified in muscle, but not in liver extracts. Also, insulin receptor substrate 1 is an upstream effector of insulin receptor activation, and increased phosphorylation of insulin receptor 1 in skeletal muscle extracts were found in AKAP150 knockout mice. In addition to these proteins, another protein was found to be more active in AKAP150 knockout mice. That is AMP-activated protein kinase, which is known to regulate glucose metabolism. It was found with immunoblot analysis that skeletal muscle and not liver shows increased activity of AMPK. Therefore, it is concluded that increased insulin sensitivity in skeletal muscle is an adaptive response to reduced serum insulin levels in mice that lack the AKAP150 gene. The point of this experiment is that these anchored proteins can be used to create new drugs that can fight effectively diabetes. Research has used imaging techniques combined with genetically modified insulin cells and whole mice.
Mice lacking alpha kinase protein 150 didn't produce as much insulin from beta cells in pancreas, but they had better insulin sensitivity in skeletal muscles. Scientists, therefore, were able to show glucose control came from 7 amino acid sequences in the anchor protein that directly interacts with the surface of phosphatase enzyme.